I appreciate y'all being here. This is the first of a new science webinar series that we've started with the goal of disseminating our science information, both within our agency and ultimately to the general public as well. So we've got over 50 people in, and our first webinar is going to be by Jake Rash, and he's going to talk about the foundational development of the brook trout genetics in North Carolina. And I will say, if you have a question as Jake's going through, if you will, type it in the chat box. And our plan is to have him go through those questions at the end of the webinar. And so, Jake, are you good? Are you ready? If you are, go ahead and share your screen and take off.
I am ready, David. Thank you. All right, I believe that's presented. Yeah, David, thank you for that introduction, and certainly appreciate the opportunity to be here today to chat with everyone about just some of the exciting work that we're doing to help conserve brook trout across North Carolina. And truthfully, this work is also exciting because it contributes to the greater efforts across the native range of brook trout to help them in all the places that they're found. And so I'm going to take time today I'm definitely going to talk about the science, but I want to take today to really get into sort of why we're doing the science, but also what it means and how we use it, how we put it to work to help us make those decisions. And so we'll work through here. And as David said at the end, I'll be glad to take any questions as they come to you. And I look forward to sharing this with you. And I hope you enjoy it. But before I really get started, I want to put you to work. Take a second. Think about the mountains. What comes to mind for you when you hear that word? Like, what do you think about? I mean, it could be something as as iconic as black bears that are really associated with our mountain region. It might be where you live. It might be places that you go to visit. It's some cherished times with family, maybe like here, getting out on the water, just enjoying the beauty that's really tied to this area. Here's a picture of the Blue Ridge Parkway. Sort of its iconic vistas with the mountain foliage here. And it is just different than some of the other places that we spend a lot of our time with the hustle and bustle, and it's a place to kind of get away or to think about all these other images and things that kind of draw us to it. But my goal today is, when you think about mountains, or if you're driving through the mountains to go somewhere, or you're planning your trip, that you might actually think about this fish, and think about how it's truly special to us here in North Carolina. And part of that, uniqueness is it's our state's only native trout. And it's certainly pretty, as you can see from the images here, particularly in the fall when those males get those really bright spawning colors. So it has this just really inherent biological and ecological value to our region and to our state. But that's not all. I mean, if you're here in the mountains, or if you're familiar with anyone with a connection to the region, you've probably heard a story about specks, which is what a lot of local folks refer to brook trout, a speckled trout, or specks. And that long-standing tradition of growing up with these fishes in these hills is deeply rooted, in that fabric of our mountain communities, and that's not lost through time, and that continues today to current generations, and that sort of value and and the utilization and appreciation of these resources. And I can't tell you the number of times we go to knock on the door to ask for permission to sample the stream on somebody's property. And once we tell them what we're doing and what we're looking for, those stories start to flow about those fish that were up in the hollers, or somebody from generations ago that had something to do with them. And that social connection is really strong through today, just as I noted. And so I want to expand on that. Here's a depiction of a watershed, and when we think about brook trout, they're up here in the headwaters. And I just went through a series of reasons with their importance for communities and the ecology and biology up here in the headwaters. But if we think about it, they're at the very top of these watersheds where that water is flowing downstream to where many of us live. And brook trout require such pristine conditions. They need really clean, really cold water to persist. So if we're doing things that can benefit these populations here in the headwaters, that trout conservation, those efforts really multiply and flow downstream to the species that are adjacent to them and even further downstream, but also to us where we're living in these cities. Not only we're living, but we're recreating in these waters. And so we're able to do this work and to think about brook trout conservation. And we're obviously working to help these populations at a local level. But at the end of the day, that work has a chance to really impact things that are downstream, including us. So think about that as we move forward and we think about these fish. Well, speaking of impacts, conditions weren't always great for brook trout in our mountains. Really intensive logging practices in the real late 1800s period saw pretty extensive impacts across the landscape. And this picture on the right, you even see, this wasn't North Carolina, but it depicts how waterways were used to transport timber out of those mountains into the lower elevations or lower reaches where they could be processed. That had a significant effect across the North Carolina mountains and what it did to populations at that point in time. But not only that, we also have the introduction of non-native trout species to North Carolina. And that really started in the late 1880s with the introduction of rainbow trout from the western United States. And then in the early 1900s, we saw brown trout come over from Europe. And since then, both have established self-sustaining populations throughout North Carolina and really the United States. But not only that, we saw an influx of hatchery stream brook trout. If you think back to those impacts I showed you a couple slides ago, folks really started to notice that these pretty little fish that were here, they're not here anymore. And so they wanted to do their part and try to get them back. Well, to do that, they brought fish down from the north. And really, any hatchery brook trout can trace their origins back to hatcheries in the Northeast, in those New England states. So those fish with hatchery genetics were brought down to North Carolina, and then they were spread out and continued to spread out, and then moved all across the mountain region. And so the dispersal of those hatchery fish was extremely widespread. So for us, we do a considerable amount of work to try to find populations of brook trout. And so what you see depicted here is what we know today of where our brook trout populations are or have been found throughout western North Carolina. And if you're not familiar, this is the process that we use to sample trout. It's called backpack electrofishing. And it allows us the opportunity to collect individuals, do what we need to do, weigh, measure, collect genetic tissue, and release them back into the stream. But when we think about this brook trout distribution in a little more detail, we can see some patterns that really stand out. And the biggest of which is most of these populations are over 3,000 feet in elevation. And that's significant for several reasons, but the one of which I'll talk quite a bit about more here later in the talk when it comes to genetics. In 3,000 feet in elevation, it may not sound like a lot, but when you're trying to get to all these different places to locate these animals, it can be a challenge. And so let's just talk about that real quick. We'll go back to our watershed example, and this red line, for our purposes, we'll think of that about that 3,000 foot elevation mark. And so what does a typical barrier look like? Well, we're really fortunate to have advances in technology like Google Earth, where we can sit on our desktop and kind of scroll up a stream to get a sense where a particular barrier might be. So let's go through this, and we'll walk our way up a stream channel. And we're heading up, and then take a look right there, and then you see it flattens out. That point that I showed you right here in real life is that. These types of barriers are really spread out and consistent all across our mountains. And we see real examples like this where that's a barrier to prevent fish movement upstream. But if you remember from that Google Earth video, it sort of plateaued once you got past this. And sure enough, in real life, once you got past this fall or slide, we find a brook trout population. And so that's a common story throughout our mountains, and one that requires a lot of work to investigate and to make sure that we're not missing potential populations. So this is right. Yeah, it took a lot of work to find them. And so that's good. We're sort of locating them, and any day we find a new brook trout population is an awesome day. But really, it gives us the chance to ask more questions. We've got them, but what in the world are they? And that's where our genetic work can really come in and help us further explore these populations and answer a considerable amount of more questions than we would have otherwise. Well, that genetic work, our genetic baseline, it really got started with in-depth work with allozymes, which allozymes are a variant of a protein, and in brook trout, it's the creatine kinase protein. And so what it does is it allows you to tell the difference among that protein and determine any influence of those hatchery genes, those hatchery stockings I mentioned earlier. And through that initial allozyme work, we did about 500 populations, and you can see here almost 62% of those showed some significant level of hatchery influence. And the last round of that allozyme work was conducted back in 2005. So again, a tool to help us determine that hatchery influence. Well, fast forward a few years, we're able to advance those techniques and use something termed microsatellite loci. And in this case, we use 12 microsatellites to take a look at our individuals. And this contemporary form of genetic analyses really gives us a more robust understanding of our population's genetics and the relationships between and among all those populations. And we're really fortunate to have close to 500 collections and 9,500 individuals been analyzed through this process. And that's really one of, if not the largest, state-level data sets that exist. So we're certainly fortunate to have those resources. And so we're really fortunate as well to be able to use tissue that was collected previously from those allozyme analyses, but also we're collecting untested populations as well. And so this is great because it not only helps us here in North Carolina, but it lets us compare our fish with those throughout the range as well. And so again, that's helping us contribute to this better understanding of what we're seeing here in North Carolina compared to what they're seeing throughout the range, whether it's in West Virginia, Pennsylvania, what have you. But this also allows us to take a look at 15 hatchery collections as well. So we're also able to take a really deep dive into that question of hatchery integration into our populations as well, similar to what the Alazine work did, but at a much finer scale. So there's a bunch of different data that can come out of that work, but today I really just want to focus on two things: genetic diversity and hatchery integration. But before I do, I want to spend a little bit of time on genetic diversity and how I think about it. I sort of think of it as a group of tools relative to our toolbox. You know, we've got a toolbox with a diverse set of tools, and at the end of the day, genes are acting like these tools. Each one has sort of a different function and a different role, and you can get deeper where there are alleles, which are variants of genes that also, you know, are much finer scale. But for today's talk, we'll focus on genes. And so if we think about genes as each one of these different tools, they're doing different things, and we're building a toolbox that's pretty diverse, which, you know, because you never know what you're going to need a tool for, and you want your toolbox to be as full as possible to handle what comes up. Well, inevitably, you've seen the trident by now. I don't have a trident in my toolbox. That would be pretty awesome, but it's just not going to happen. And the reason I want is that it doesn't seem practical, and there's no reason to have it. So in this scenario, if we feel the same way, like, you know what, we don't need this, let's just get rid of it, and we're left with this existing group of tools in our toolbox. That's fine. That's absolutely great. You'll never need that trident. Hopefully, if you're working on your house, you've got all these other tools at your disposal. And again, think of these things in terms of genes. You've got all these things at your disposal. You're able to do what you're able to do. But what if you learn one day you're Aquaman? Well, you've now gotten rid of your trident, and Atlantis is in peril. And so the point here is you kind of never know. And that's why it's important to maintain that genetic diversity. And that was kind of an over-the-top example, but if we pull this back to the real world, we should think about things like this. Like, what if one of these genes is responsible for helping with thermal tolerance, helping individuals and groups of individuals and ultimately populations deal with changes in temperature? Or what if one of these tools is responsible for helping promote disease resistance? So a group of these individuals is able to resist a disease, whereas the others aren't. And so these three here are able to persist and pass that gene on into the future. And so that's the important part about this diversity, is you have these genes and these tools at your disposal at the individual and population level when these sort of selective pressures arise. A little bit of data here. I don't want us to get bogged down in this, but this is presenting some of the genetic diversity data. And I'm going to present you three different metrics that measure genetic diversity. And this one's the first is observed heterozygosity. And this is presented across watersheds. And the real take home here is this is actually really low compared to what it would be across the native range. However, you do see some outliers here with savanna and New River basins presented. So why would this be low? Well, it gets back to this. If you remember this slide where we're finding these populations, they're not connected to each other. They don't have what's termed effective gene flow. They're not able to exchange genes between or among those populations. And so that's what's driving our lower genetic diversity here. Well, let's think about hatchery influence. What are those hatchery genes doing across our populations? And we were able to do that through three different metrics that you see here. But what's important is down at the bottom, where we were able to combine those into a single composite score. And the the good news is we have a lot of collections closer to 1, which is the closer you get to 1, the less of the hatchery influence that you see. And so that's really encouraging. And that's also sort of opposite of what we saw with those Alazon data. If you recall, about 62% of those showed a significant level of hatchery influence. And that's part of the ability to do further investigations with this type of genetic data to get a more diagnostic approach into answering this question of how much influence hatchery genes are having. And let's go back to our watershed scenario, and I'm just presenting one of those metrics, this core distance here on the y-axis. I could have shown any of those 3, and they really all behave the same. So please don't get hung up on that. And what we see out of our major watersheds, the outlier here is really the Savannah Basin, where we are seeing higher levels of hatchery influence than the others. And if you recall, a couple slides earlier, we saw higher genetic diversity in this basin as well. Well, that's likely due to this, where we've got those influx of hatchery fish and those hatchery genes that are showing up in those populations. So I hope you agree with this fish. I think it's super cool. I just love talking about it. But it could be the neatest thing in the world. But at the end of the day, what in the world are we going to do with all that information? And I think that's a pretty fair question. And really, for us, it gets back to sort of the core of our trout management. You can see our trout management plan listed here, and you see all the genetic collection sites that we have, and we have at our disposal. And you know, the purpose of our trout management program is, you know, we're going to use science to make the best decisions that we can for our populations, and that's really, you know, some more of the exciting parts to this work. You get to research and answer questions, but then you've got to apply it. So I want to walk you through several scenarios of application and how we actually put the science to work. And one of the cool things we can do is look at all of our populations at once. And so here is another one of those diversity metrics, rarefied allelic richness, versus our composite score that we've talked about previously. So x-axis, genetic diversity, y-axis, hatchery and aggression. And so here are all of our populations plotted at once. So we can start to partition these and look at individual collections versus a whole. And so in this case, maybe we're thinking about doing a restoration with a population, meaning we want to restore a brook trout population somewhere that it was lost. And so we can start to look at all these collections and get a sense for how they're sort of behaving. For instance, like in this lower left corner, these populations have low diversity and they're pretty impacted by stockings. And so if we keep moving forward, things are a little bit better as we get out here. We've got higher genetic diversity, but again, we're still seeing that level of hatchery influence. Much better scenario as we move up and we look at this upper left quadrant where we've got low hatchery influence, but slightly less genetic diversity. And things really improve over here in this upper right corner because we're seeing sort of the best of both of these worlds. Little influence from stockings through history, and then high genetic diversity. And these are sort of the best when it comes to thinking about populations to pull fish from to move somewhere else. Well, let's expand that even further into an actual real world example here. And this is a case where it's a great case study for several reasons. One is the partnership aspect, and that's with the North Carolina Forest Service in this case. And it also speaks to the value of habitat. You can't have any fish population unless you have their habitat. And again, if you think back to the slides that I started out with, the significance of trout habitat and the specific requirements that it needs are things that that are there's paramount to the persistence of trout populations, particularly brook trout. And in this case, through a decades-long process, the NC Forest Service was able to restore trout habitat on a stretch of Palier Creek. It's just absolutely awesome. Degraded habitat brought back. And it's the Field of Dreams type thing, where if you build it, they'll come. And our part is to help to bring those fish there. and to do that, we wanted to restore a brook trout population back into the stream. And if you look on the right, and we'll this is our Huck 10 watersheds, but you can see potential places where we'd pull fish from, and then Palier Creek itself. And we'll blow up this chart a little bit, and once again, just to walk you through, these are potential source streams under our consideration. Or HOO is observed heterozygosity. Again, our genetic diversity. INSA-B is effective population size. We haven't talked about that. And then again, our composite score that we've mentioned. And this relative abundance is a metric that we came up with just to make sure that we're not taking fish from a place that can't sustain it. I mean, the last thing we want to do is do harm to a population when we're trying to help another. And so at the end of the day, we were able to look through these candidates and selected two options as potential source candidates to restore the stream. And by doing that, is sort of an opportunity for us to evaluate that approach where we're using two source populations to try to restore a population. And the initial results are positive. And this is just the census data, being able to look at the individuals, but the additional genetic work will come to see if we have been able to maybe enhance that genetic diversity within this single population by having combined those two to bring fish back. Really exciting stuff. Again, from the opportunity to restore habitat, to investigate approaches that help us better refine how we restore these populations. Rather than just throwing things against the wall, we're monitoring and evaluating these and making sure that they do stick, rather than just putting it to chance. Well, another great example of how these data are used is identifying priority habitat restoration projects. As you might guess, there's no shortage of needs to improve habitat across Western North Carolina. And this is a case where we're able to work in conjunction with additional partners, those at North Carolina State Parks, and also at Trout Unlimited. And so we can use these data to help filter through and find populations that we can isolate out and then focus efforts and energies on to try to restore them. And in this case, what you see on the left is a perch culvert that is depicted below, and what it did was it prevented fish from being able to move upstream. And so, talking about gene flow, it's only one direction, so fish can only go downstream in this case. That's not ideal, and we'll talk more about that here in a second. And you can see the after, just awesome. And so what has been able to happen here is once that culvert's removed, we've established passage, and now these fish that were stuck downstream now have the chance to get upstream. And so gene exchange is not just one direction, it's both ways now. And that's critical for the long-term health of these populations, particularly with the gene exchange, but additionally, you can think about opportunities to access more available habitat, whether it's thermal refugia or spawning locations. Again, it opens up more habitat for these fishes. But if we think about gene exchange, we can come back to our toolbox. And these two groups of fish that were previously above and below that barrier, they're kind of like neighbors, but instead of a culvert, maybe it's a big giant fence where you couldn't get over to borrow your neighbor's tool that you needed. and so that you were kind of stuck. But now we're able to really enhance the size of the toolbox of each of these groups of fish in this stream by allowing them to freely move back and forth and to freely exchange genes with each other. Really, really neat stuff. So what's next? We're honestly, we've done a lot, but we're still working really hard to expand and strengthen this genetic baseline. And we're working with our partners throughout the range and throughout North Carolina. I mean, truthfully, at the end of the day, the issues that face brook trout do not really understand administrative boundaries. And so our ability to work with partners and to work throughout the range helps the species, and not just North Carolina, but beyond for sure. And we're continuing our restoration efforts. The focus on habitat it's so critical. I mentioned earlier, you've got to have the habitat to have the fish, and things like working with our partners, that help distribute and construct information like on the right, but certainly trying to prevent things like you see on this lower left, trying to prevent that degradation of habitat, but being able to zero in and focus on those key areas to do that work. And we're certainly working to restore populations as well. It's a really good day when you can find a brook trout population, but it's also, in many regards, it's just as exciting when you, or maybe sometimes more, when you can bring one back. And so we do quite a bit of work to continue this plan and using these genetic data to help us determine where, how, and when we're going to do restorations. And if you're interested, there's an article back in the fall 2018 Wildlife in North Carolina that speaks more to how we use these genetic data to do restorations. And then also, this can help us with future land protection efforts. We're able to get a better understanding of these individual populations, and that can help us identify what could be critical parcels to the long-term success of these fish and to certain population. And we can work with all of our partners to help identify and address those needs as we go forward. And so again, we're able to really fine-tune where and how we're looking at what those potential needs might be. So at the end of the day, we have low genetic diversity in our populations because there's really little, if any, gene flow, between or among them. And the good news is the hatchery integration seems to be rather low, particularly lower than maybe we once thought historically. And the real take home is these genetic data help us prioritize limited time and resources. We can't be everywhere all the time. We do not have all the money in the world to address these issues, nor do we have all of that time to do it. So this helps us really prioritize and focus our work efforts to get to places and in manners based on these available data and best science. And at the end of the day, we're hoping that we can conserve these populations to where they're all fishable. And that's a goal that we want, because we want future generations to always enjoy these animals. And this work doesn't happen without a ton of people's help. I'm just fortunate enough to be here talking about it, but it's really everybody else that's been involved that has made it a reality. And the folks here have inevitably been out sampling a brook trout at some point in their careers. And then a few of them are part of our brook trout crew, which is comprised of a 2-person seasonal crew that they go out and they hunt trout populations, but specifically brook trout, and collect genetic information. And they're the ones that are really driving a lot of this work. And here's a picture of Kenneth Lingerfeld here, which this was on the cover of the Asheville Citizen-Times a couple years ago, and really neat that this got picked up and circulated around the country. But certainly wish to thank all of the folks that helped on the genetic analyses as well. At Western Carolina and Auburn University were instrumental in those allozyme analyses initially. And then the folks at USGS have really shepherded us through the contemporary work with the microsatellites. Dr. Tim King is certainly a visionary and someone who was a champion for doing this work and doing it with brook trout. And he got us started and really paved the way for us and many others in the field of brook trout genetics. Unfortunately, Tim passed before he could see, much of this work reach sort of the culmination of things like I've showed you today. But his legacy and impact will live on as long as anybody's looking at genetics in brook trout. And with that said, we thank Dr. Dave Kazak, Barb Levinsky, and Dr. Steve Faulkner as well at USGS for really helping us carry on and continue all that work and to keep pushing the envelope, keep learning and keep striving and collecting these data to help us learn more about our brook trout. And none of this happens without multiple partners, but I really want to thank the land managers that are out there. Certainly, we're fortunate to have the two large forests of the U.S. Forest Service here, and certainly North Carolina Forest Service, the Park Service, state parks, all the municipalities, in particular the city of Asheville and the Asheville watershed, the Land Trust community with all the work they do to conserve lands across western North Carolina, and private landowners. They play an integral part in this discussion, particularly if you think about the Wildlife Resources Commission District 7, which doesn't have a lot of public land, but they have a lot of brook trout. And it's through that cooperation and stewardship of those landowners that those populations are able to have those conservation measures that they receive. So we're certainly grateful. So I've done a lot of talking. If you're still listening, I really appreciate that. But I wanted to show you something that to ease the pain of all my talking. But I think sometimes it can get lost. I mean, what we're really referencing here. I mentioned data, some numbers, some pictures, but I think at the end of the day, it might help us to take a look specifically at the world that we're referencing here and look directly at the fish themselves. So I'm going to play a short 2-minute video. There's no sound here, but this is some really impressive footage that was taken by Kenneth Lingerfeld that he agreed to share with us, and I want to share it with you to help us again to think about what we're actually talking about here. So I can't thank Kenneth enough for his willingness to share that and to let us have the opportunity to see that video and that footage. That is really unique, and there's not a lot of video out there like that shows those spawning behaviors, particularly here in North Carolina. But I hope that gives you a sense of what we're talking about here, just the splendor and the beauty of those fish. And then what we were seeing are the stages of gene exchange in action with all of that spawning activity and things that take place. And it shows it's kind of complicated, and there's a lot that's happening in the water just to have a successful spawn. But it gives us a sense of, you know, for me, the beauty and what we're really talking about here. And again, I appreciate Kenneth being willing to share that footage. And so if we go back to this slide again with the watershed, and we think about, you know, those fish we just saw in that video, and we think about where we're at in this grand scheme of things of a watershed, and we think about the interconnectedness of nature and how things are connected. And again, it's thinking about that work that we're able to do for these species and using those genetic data to help us make the best decisions we can to conserve these animals. And as we do that, we're thinking about that trout conservation flowing downstream to all of us as well. And so I hope you think about that the next time you think about the mountains, or like I said, if you're driving through, or if you see a trout, particularly a brook trout. But I hope at the end of the day, you've been able to get a glimpse of why we're doing the work that we're doing, but also how we use the science to help us make those decisions to conserve trout. And with that, I'll conclude, and I certainly am, again, appreciative of everyone's time. I know everyone is busy, so thank you for taking time out of your days to listen to this. But I'll take any questions now if time allows. And obviously, if we're not able to get to them today, or you think of something later, feel free to get up with me at any point. If the last 40 minutes or so haven't made it clear, I love to talk about brook trout, and so I will be more than happy to do that at your convenience. But thank you again for your time.
Jake, that was great. Thank you. Thank you for doing that. Thank you for taking not only the time you've taken today, but the time that I know you've taken over the last few weeks to put this together, to not only explain the science that's going on in the cold water research world, specifically with brook trout, but also to very clearly connect that to the conservation work and to the things that are important, not only to the 58 or so people that held out with you for the whole time, by the way, because I did keep track, but also to the folks in North Carolina. Let's see, something just popped in. Let me see here. So there's one question here, Jake. How concerning is the loss of hemlocks to brook trout habitat?
Yeah, that's a really good question, and one that's kind of an emerging, topic and field. There's work ongoing to sort of answer that question, and it's really about, what species are going to come in behind them to replace them, to help maintain whether that's the bank stability, but in particularly the shading. So that's definitely a good question. They are providing woody impacts or woody inputs into the systems, which are critical to diversify habitats, and so it is contributing positives in that regard. But what comes in behind them, and how soon and how fast, that's sort of an emerging piece.
Let's see. So Jake, here's another one. let's see if I can get it all. Do you work closely with the Great Smoky Mountains National Park? Are they doing genetic work in a similar way that you can use to augment your genetic assessments?
Yes, 100%. That's one of the great things about this work, and one of the great things about trout management and the various agencies here in the Southeast. We all work together closely, and we work very closely with the Smokies, and then of course really everyone else as well. And so we're sharing those data, we're combining our data, and in fact, any analyses that we're doing, when we're reflecting that against larger spatial scales or throughout the range, it's capturing their data and vice versa. And so that's something that we do regularly, and speak and work with them a lot, and all other trout resource managers in the region as well, and really the Northeast as well. So throughout the native range.
Okay. And another one is, do you have a feel for how many streams are available for restoration similar to the work in Wilkes County?
Um, a lot. Numbers wise, that's I don't have a specific number, but there's a lot, and there's several factors that go into determining what is suitable. And so that's where, going back to the previous question of partnerships, that's where the partners really can come into play, because when we're working with folks like the Blue Ridge Parkway, or the U.S. Forest Service, or the NC Forest Service, or NC State Parks, or any of those groups of folks, we can work with them to help drill down and identify more of those priorities, and that can tie into planned or existing habitat restoration projects, or it could be relying on their institutional data or their knowledge of where potential opportunities might be. And then we can also partner and work with them to help meet their objectives too. So I don't have a number, but there's enough to keep us busy for the foreseeable future.
Okay. Do you know if anyone is doing genetic work to capture adaptation for climate change? For example, increased water temperature tolerance. And if so, is any of that work relevant to North Carolina?
Yes, Dr. Than Hitt up at USGS Leetown is doing some work with gene expression, looking at thermal tolerance in trout species, but brook trout species. And so he's a collaborator of ours on a lot of other projects and efforts that we're working on. And so we're keeping an eye on his work and looking to hear more about it. And I think there's more work out there too that's coming. But no doubt we're on the southeastern range of trout populations, and so we're always looking for, one, to see what's kind of interesting research is out there, but two, as folks are starting to take sort of the next steps and really looking more into how those genes are working and which genes in particular are doing what. That's pretty cool, because that's going to be even more tools to put in our knowledge toolbox to keep that analogy going.
All right, let's see. Along the lines of the hemlock question, do you see an influence from pH changes such as our vegetation changes? Do you see an influence from pH changes as our vegetation changes, such as encroaching rhododendron?
Yeah, that's a really good question too. The folks at Coweta have done quite a bit of work of that, the Coweta Hydrological Station, and yeah, and that's another one of those that they've done work, but that's kind of again part of that whole kind of the welding the wedding of the forestry discipline with the aquatics and hydrology disciplines to learn more about that. And to be honest, I'd have to dive into that question a little more, but it's definitely something to give you a more definite answer, but it's definitely something that people are concerned about and folks are looking into. And from our perspective, again, that's one of the beauties of having strong partnerships where we can kind of rely on that work that our partner agencies are conducting, and to get a better sense of that. But certainly at the end of the day, when you look across that landscape, and it's back to that notion of things in nature being connected, right? As these vegetations potentially change, and what are those potential influences? That's something that we will want to keep our finger on.
Very good. That's the last question we had. We're about let's see, hold on. Nope. All right, I'm gonna give you one more. How are we keeping non-native trout out of native streams?
Yeah. Yep. That's a really good question, and a challenge. There's a couple ways. Let's start with ourselves. We have a policy where we're not stocking on top of wild brook trout populations, and in addition, the fish that we stock are sterile, and so we made that switch over a decade ago to ensure that any fish that we're putting out is going where we want it to go, and should it get somewhere unintended, it would have a punctuated life cycle and it wouldn't reproduce. And so that was really a move to help us protect our native brook trout populations. And so that's us. On the other side of things, North Carolina is second to Idaho in the amount of trout produced. That's I say that number and it's still kind of hard for me to believe, but it's there. So there's a lot of fish being produced and there's a lot of fish being brought in. We require anybody stocking waters to have a stocking permit, which comes through us, and we're primarily reviewing those to make sure we don't get those fish into our native populations. And so that's our mechanism, is that through those stocking permits, and those are dependent upon folks getting a permit. And so we definitely encourage that, but it's hard to get 100% compliance. So those are the main ways that we do that. And third, when we think about those habitat projects, we sometimes think about some barriers as positives, because we may not want to remove a certain barrier because it's keeping those browns or rainbows out of a brook trout population. So we'll actually advocate to leave those in that instance to provide further protection.
All right, Jake, I think I'm going to call it off there. That's the last question in the list, and we're right here at noon. I'm going to thank you again, and thank everybody that was on the webinar. I will put in a shameless plug. This is the first of what, as I said, of what's going to become a series. So if folks have any suggestions on how to improve how we do this, the process, any of that kind of stuff, if you will, email that to me. And all the accolades for how great Jake did, you can email to him and just copy me. And again, I appreciate it, Jake, and I appreciate everybody else.
Yep. Thanks, everyone.
See y'all later. Be safe.